Hello, welcome to UCF and the UCF Chemistry Department. I'm Sherry Yastrebski, Pegasus Professor and Department Chair of Chemistry. The department faculty, staff, students, and myself are all very happy to have you visit our university. We have a young university, established in 1963 with groundbreaking in 1967. At that time, we were known as Florida Technological University, with a name change in 1978 to University of Central Florida. In 1978, we had a student body of about 11,000. In fall of 2018, we had an enrollment of about 68,000, making UCF the second largest university in the country. The chemistry department was established in 1967 with an ACS approved bachelor's program in chemistry in place by 1969. In 1978, a forensic science bachelor's program had begun. Today, that forensic science bachelor's program is ACS approved and accredited through the American Academy of Forensic Scientists. By 1978, we had developed a master's program in industrial chemistry. In 2005, we started a doctoral program in chemistry. Today, we have 42 very active faculty directing the research of over 100 graduate students in areas including chemistry education, biochemistry, environmental, materials, and forensics chemistry. Our faculty bring in several $million of funding every year and publish well over 100 peer-reviewed publications in highly regarded scientific journals. I hope you enjoy perusing some of that research, talking to our students and faculty, and touring a bit of our campus.
Hello, my name is Yulia Gerasimova and I am an assistant professor at the chemistry department at UCF. In my lab, we use DNA as a tool to study or analyze other nucleic acid sequences. And the probes we develop follow a unique multi-component approach that makes them multifunctional. They can tightly bind to even highly structured nucleic acid target. They can differentiate between closely related sequences. They can do so even in complex samples such as in cellular extracts. They can use either fluorescence or electrochemical signal or color change as a readout. And color change is a particularly attractive signal because now you can detect the presence of your nucleic acid target of interest without the need of expensive instrumentation because the signal can be observed with the naked eye. So we are using our probes to detect pathogenic bacteria and viruses for point-of-care diagnostic tests. And we are happy to provide our probes for other important biochemical tasks. Hope you enjoy your visit at UCF today. Welcome here.
Hi, welcome to UCF. I am Fernando Uribe Romo. I'm an assistant professor here in the chemistry department, and we work in an inorganic chemistry lab where we create new materials for sustainable use of energy. The materials that we make are called metal organic frameworks, and are materials that combine the properties of organic molecules with inorganic crystal structures. We make our materials to find applications in uses such as catalysis, solid-state electrolytes, gas separation, gas storage, carbon dioxide sequestration, and in general, renewables, sustainable use of energy. Thank you very much for visiting UCF, and I hope you enjoy your time.
Hello, my name is Jack Ballantyne. I'm a forensic scientist and a professor of chemistry here at UCF with a joint appointment at the National Center for Forensic Science. Forensic science is an applied science that helps establish facts that may be in question. These facts pertain to criminal investigations or other medical legal matters, and the questions revolve around what happened, where did it happen, how did it happen, when did it happen, etc., with a very important part of that being who caused it to happen or who did it happen to. That's the identification side of things. DNA profiling has revolutionized forensic science because with DNA profiling now it's quite routine to be able to obtain the genetic profile and identify an individual based upon a biological trace left at a crime scene. My lab uses, my group here uses techniques and technologies from biochemistry, molecular biology, and genomics to help forensic scientists retrieve more information from small amounts of material transferred during the commission of crime. And we take a problem-oriented approach to our research. And the three thematic areas that we're working on right now are activity. That is, what activity led, what happened? In other words, remember the questions. What happened when this sample was transferred at the crime scene? That can be very important. It can be very important, for example, in sexual assault investigations. So the sexual assault situation, we can get a DNA profile, but how did the DNA profile get there? And the question is, was this due to social intercourse versus sexual intercourse? There may be an innocent explanation why the DNA profile was transferred or not. And discovering biomarkers for saliva, vaginal secretion, skin, for example, can help the forensic scientists ascertain which of the two scenarios did occur. So we've discovered some of these biomarkers, and they turn out to be RNA and not DNA based. And we've helped establish, in fact, this burgeoning new field of forensics, RNA forensics instead of just DNA forensics. That's the first theme. The second theme is trace evidence analysis. Trace evidence analysis is a situation whereby we use single cells to help with mixture analysis. Mixture analysis is the situation whereby body fluids from more than one individual are transferred, and you get a mixed DNA profile. Sometimes it's difficult to deal with that. So by performing single cell analysis, which requires enhanced DNA typing methodologies and micromanipulation methods, we can remove single cells, either epithelial cells or single sperm cells, and help deconvolute that mixture. So this will be a useful addition to the forensic scientist toolbox. The third area, third theme is sexual assault investigations, in which we have helped the community, the victim community, to obtain DNA profiles from victims who've come and reported the case more than 3 days after the incident. So typically a standard DNA analysis, after 3 days, you can't get a result. But with the methods that we've developed here using Y chromosomal methods, we can get results 5, 7, and 9 days after the sexual assault. And our work has featured in the reason why the Department of Justice has recently indicated that sexual assault evidence should be collected 5 days after the incident and not just 3 days. So we're quite proud of that. So with that short introduction to our research, I should like to wish you all the best for your visit here at UCF.
Hi, I'm Erin Saida, and I'm a chemistry education researcher. My work focuses on implementing evidence-based teaching practices in STEM courses. An area of particular interest involves how we prepare our graduate teaching assistants for active learning environments. My research team is currently investigating the use of a mixed reality teaching simulator to create rehearsal opportunities for GTAs to practice high-impact discourse strategies to make their teaching more effective. I welcome you to stop by and talk to me if you're interested, and I hope you enjoy your visit.
Hi, welcome to the Beasley Group here at the University of Central Florida in the Department of Chemistry. In our group, we study environmental chemistry and microbiology. Specifically, we study the fate and transport of contaminants in our environment, such as hydrocarbons resulting from oil spills, heavy metals resulting from some anthropogenic release and contamination, and even up to sucralose in personal care products, as well as pharmaceuticals resulting from just our everyday human use. We're very concerned with the speciation of these chemicals and these particular contaminants, because it's the speciation that's going to tell us how mobile or how persistent they are in our environment. And they can also lead to help us develop mitigation as well as bioremediation strategies. On the other side, we're also very interested in microbial ecology and also these particular contaminants and how they affect the microbial community of these particular environment. And so for doing this, we use next-gen sequencing as well as culture-dependent methods for studying these. Thank you, and have a good time at ACS.
Whether you are an alum of UCF or here for the first time, I think you will be impressed with the campus, the facilities, and people who make our university and department so engaging and productive. Welcome to UCF.